Welcome to On-Site Insights, a series of podcasts by On-Site Ventures about deep tech and the people behind it. We invest in the next groundbreaking idea.
Welcome everyone to our 7th live session of On-Site Insights. On-Site Insights is a series of LinkedIn audio events with the goal to bring our portfolio companies and the people behind them into the spotlight. My name is Klaus. I am a general partner and founder of On-Site Ventures, a venture capital fund dedicated to invest in early-stage deep tech companies. Today, I have the honor to have Miroslav Gasparek, co-founder and CEO at Sensible Biotechnologies, as my guest. Miro and his team at Sensible are working on the next generation of mRNA medicines. The in vitro production process of mRNA is highly challenging and costly one. And with in vitro production of mRNA, it is still limiting the therapeutic potential. Here, Miro and his co-founder Marian Kupchalak come in with Sensible. They are working on a novel mRNA approach. Thank you, Miro, for being with us today. You're a well-known founder. You have been featured in the Forbes magazine and have raised a substantial pre-seed round from some of the best VCs out there. Welcome.
Thank you very much, Klaus.
Can you start by telling our listeners a bit about yourself and your way into the startup space?
Absolutely. So I'm originally from a little village in northwestern part of Slovakia, where there, as you can imagine, wasn't very much biotech. And throughout my youth, I was always very passionate about two areas of the you could say science and medicine. So it was a theoretical astrophysics and theoretical physics on one side, and actually the medicine on the other side. And I was kind of hopelessly torn between what I should do. I wanted to combine the quantitative understanding that the physics and astrophysics were providing with the ability to help people through that quantitative understanding of the processes in a biology. And that led me to study biomedical engineering at Imperial College London for my undergraduate degree. And I was very lucky that very early on I started engaging in a research in systems biology and got opportunity to work on that by Professor Eiko Tanaka. And I started working on eczema treatment, on a modeling optimal treatment for eczema. And that brought me to control theory and subsequently into synthetic biology. And I was again lucky that I got to work with Richard Murray at Caltech and subsequently with Drew Andy at Stanford later in my undergraduate career. And especially the time at Stanford was transformational. Everybody was vibing and everybody was essentially living by next big entrepreneurial idea. And I thought, wow, this is what I want to do. I want to be kind of scientifically apt entrepreneur, an executive who will be helping to build the next generation life science companies and will help people through innovation. That's why then subsequently I ended up going for my PhD at Oxford and the PhD in engineering and synthetic biology and kind of combination of nonlinear systems modeling applied synthetic biology. So that was kind of a really good base for my kind of broader thinking. And I had fantastic supervisors, Professor Andres Papachristou and Professor Harrison Steele, who from the beginning of my PhD career just basically said that they kind of were tolerant to my entrepreneurial ambition, and essentially then soon after I started my PhD at Oxford, the pandemic hit, and suddenly, obviously, the COVID-19 pandemic was a big transformational event. Our government in Slovakia was searching for people to work with them on COVID-19 modeling and kind of analysis and so on. So I ended up working on that for a couple of months, and that brought me closer to biosecurity. And this kind of venture into biosecurity was really meaningful because I got to work with Megan Palmer at Stanford on some synthetic biology related biosecurity challenges. And that really affirmed me that, hey, could we kind of transform this problem? Like, this biosecurity in synthetic biology has multiple facets. It has this aspect of, how do we engineer the cells to not behave in detrimental way, but there was also this more global picture. How do we ensure that there is enough vaccines and access to the novel medicines for the people across the globe? Throughout the time, I was already talking and thinking about what to do with my co-founder and my long-term friend Marian, who was just finishing his PhD at Oxford in biochemistry when I was starting. And in September 21, after going through Y Combinator and essentially after pivoting for a while, we essentially decided to address the biggest problem that we thought we can address by our combined skills. And that problem was mRNA. And the excitement arising from this novel platform that saved millions of lives across the globe, that actually gave us the ability to, get out from all these lockdowns. And we thought there is much more in that than just infectious disease vaccines, but the manufacturing challenges for mRNA seemed to be immense and literally being a roadblock towards achieving or working on these novel areas of the mRNA. And that's why we started building Sensible. And, I would say that the rest is the history.
Thank you. That was quite substantial introduction. But I mean, I'm in favor that Marian is as well a great founder and entrepreneur. But maybe could you explain to our audience what you two, the founding team, bring together in forming Sensible, and what resources you're bringing into this company?
Mm-hmm. Absolutely. And I want to say, this would not be, Sensible would not be possible without Marian. He's a fantastic both friend and entrepreneur and an enterprising scientist. And I think, so both of us have Marian has a scientific background, and that gives him, I think, this systematic ability to debug and analyze the problems and be very rigorous. I have engineering background, and what we were taught to build and to fix problems. And what really mattered was that you need to essentially find a solution, almost like no matter what. And I think Marian is a very good science manager as well, able to build out that organization. And I think my kind of strong point is the ability to take that science, actually understand it in full, and with that understanding then simplify it to the external world. And I'll just quickly say something that helped me a lot in that was when I was learning from Ramsey in Nora at Civilization Ventures, with whom I worked earlier in my PhD career for a while, basically learning about the perception from the other side of the table, whether that's an investor, whether that's whether it is a policymaker or a politician, or if it's a pharma partner, that really helped me to figure out how to appropriately communicate what we are doing, because everybody kind of quote unquote clicks with something slightly different.
As you know, with OnSite, we invest quite early, so I would guess it would be quite interesting for some of the listeners who are thinking of spinning out companies from other industry partners or universities to understand how your founding story of Sensible was like.
Mm-hmm. So we are first-time founders with Marian, and we knew very little at the beginning. I think one of the biggest lessons, and I think this is where Y Combinator, and I always have to give a lot of credit to the partner Jared Friedman, helped us a lot, and also Andrei Kiska, our early angel investor, helped us a lot with, was basically really focus on the question of the problem and the market, right? Because I think as a scientific entrepreneurs or as a scientist, we very often get very excited by the particular scientific idea, whether that's, our PhD work or something like that. And then we have this quote unquote solution, and we are searching for a problem. And what we did was quite different. So we didn't in the beginning have, the technology ready to go, but we really identified and I think well-defined the problem. The problem was mRNA manufacturing. And we were able to talk to many academics, many investors, many companies in the space. And very early on, we saw that, what we are proposing is resonating with them. And I think that it was very exciting to kind of see that, you're getting this confirmation early on because, Klaus, you're actually the investor who understands this. But I think people are the biggest problem is if you end up solving a small problem with this very small market size that that doesn't necessarily can produce venture-backed returns. And I think this is something that we avoided very early on by quickly talking to the customers. And this is what YC helped us a lot with. Basically going straight to the customers and learning from them what they would want.
Mm-hmm.
How has Sensible evolved since its inception, and what are the major milestones you have achieved so far?
So essentially, well, there's been a lot. So first of all, Sensible started as just two founders in one Zoom room. Still in a pandemic, thinking about what to do. Now, where Sensible has gone by now. So we first of all, I think we built a fantastic team, which is something that makes me extremely proud. We are 25 people across the US, UK, and Slovakia. Our colleagues are the leading mRNA experts, the clinical experts, and mRNA scientists from previously at companies such as Pfizer, Merck, Medtronic, TriLink. And I think that this is very exciting, that just the sheer quality and seniority of the scientific talent is something that I'm very proud of, as well as the passion and zeal that our more junior colleagues are willing to put into their work. So other thing that we achieved, we really showed that our platform works. Sensible's mRNA platform, vision for which is really to enable and accelerate the advancement of the mRNA medicines to the clinic, which is facing challenges, most importantly on mRNA quality side, is really showing to work, and we've been able to prove our kind of core points that we were aiming to show that are kind of on the technical side. And the other thing that I'm very proud of is exactly our kind of partnering activity, not just with Ginkgo, but with other major players. And I'm really excited about the discussions with the major public pharmas and biopharmas that are excited about Sensible's technology, and also obviously about the number of other private companies that are excited about our work, and as well as academic partnerships with a network of leading scientists across the globe. And one other thing is that makes me really kind of pleased is that Sensible's other part of mission is not just accelerate the advancement of the most advanced RNA therapeutics and its next generation, but making it affordable and available to people across the globe, and to contribute to the global biosecurity and biomanufacturing resilience. And we are excited about seeing how the major national and international bodies are absolutely resonating with this vision, and about the non-dilutive funding opportunities that are kind of coming to Sensible. So I would say that these are among the most exciting, really big picture things. Obviously, there's much more, but I would say that this is kind of among the most exciting areas.
Shifting gears a little, could you explain to our listeners what Sensible does?
Mm-hmm. No, absolutely. So I think there is a couple of things that I like to say about mRNA. So what we think at Sensible is that, again, mRNA is a platform technology that can bring us much more than just infectious disease vaccines, right? And the vision for mRNA technology is to be able to provide the human body and human cells with the instructions for production of any protein that it needs, be it, missing proteins such as in, inherited errors of metabolism, be it, information that enables body to fight a cancer, or be it information about the antigens so that body can, create the antibodies and essentially prepare, have a prophylactic protection. Now, I think there are kind of two big problems with the mRNA technology, why it hasn't seen the fruition. And one of them is on the delivery side, right? Delivering RNA is challenging. It's not easy to get it into a number of organs. But I think conceptually even bigger problem and less pronounced for many people than delivery is mRNA manufacturing. Because, it doesn't matter, how well you can deliver if what you are delivering is not great. And the problems with RNA manufacturing, with this in vitro transcription-based, cell-free process, are several. But among the core problem is kind of the quality of the RNA that's coming out of that. And there's the biggest challenge that kind of mRNA assets in the clinics are facing, among the others, are the immunogenicity of the RNA, and also its functionality. How much kind of functional protein you can get out of that, say a transcript of RNA, and the immunogenicity, is to a great extent related to the impurities of the mRNA that are, still, very hard to purify and still somehow present. And something that we are aiming to do at Sensible is to address these quality problems, essentially with our novel mRNA production platform, which combines, principles of machine learning, synthetic biology, and advanced bioprocessing, where we are essentially working on extremely pure, non-immunogenic, non-toxic, and highly functional RNA. That's kind of the vision for Sensible Biotechnologies work, because we think that that's what's needed to enable these advanced medicines, gene therapies, enzyme replacement therapies, cancer vaccines, and other medicines that are based on RNA, and we wouldn't have them otherwise. But there are other challenges with mRNA production. The other challenges are related to the complexity of that production process and to the cost of that production process and the complexity of the logistics, right? To make mRNA using in vitro transcription process, you need many different, expensive and hard to source reagents, such as RNA polymerases, nucleotides, a lot of very high quality DNA template, so on and so forth. What Sensible's work is focused on as well is removing the need for these reagents. And this has a couple of implications. So one massive implication, obviously, is for the affordability and kind of the cost of RNA that you are able to get, right? Basically, we saw this during the pandemics, that mRNA vaccine was designed very quickly, say at the order of days and weeks. Then at the order of months, we're able to go through the clinical trials. But essentially, the distribution and manufacturing and, all the challenges of the mRNA, and obviously also due to the societal challenges, literally has taken infinite time because we haven't vaccinated all the people. We essentially, the pandemic ended, or the acute phase of the pandemic ended before we could even vaccinate everybody. And we think that, if you could have mRNA manufacturing facility and mRNA platform capabilities in every country, and you could locally, very rapidly produce RNA using completely novel way, which removes the need for all of these complex reagents and for all of these logistic challenges, you could really improve the access to the mRNA medicines. And this is, I think, something we should all strive for.
Great, thanks. So I think this was already kind of a sneak peek into how you would probably see a future in 5 to 10 years and the impact of Sensible's technology on the future of mRNA medicine. Well, moving a bit more into the whole industry, where are you seeing Sensible differentiate itself from its competitors, and how are the advantages of Sensible's technology positioned in these quite competitive market?
Mm-hmm. So I think, as I've said, Sensible basically has completely differentiated mRNA production platform. Our mRNA platform is kind of based on quite a different fundamental biological principles than the in vitro transcription-based mRNA production. The exciting thing about what we are doing is, I think, the leveraging of the aim to do high throughput data generation and then machine learning on that generated data to inform our biological experiments and to essentially rapidly produce better mRNA molecules. So our advantage, so what we are essentially looking at is we are making amounts of mRNA that are sufficient for any kind of evaluation partnerships or for preclinical work, which makes us really, really excited. We are making, we validated this internally and externally with the leading partners, which we think is really exciting too. And we are essentially already at a point where we have shown that our mRNA can be very, very pure, that it can have some really competitive properties to the commercial RNAs, and where we can essentially spark the interest of major or the leading public biopharma companies in our platform. And I think this is what is really important to us, because I think one thing we haven't touched on is Sensible's business model, and I'd like to kind of maybe comment on that and maybe take it a bit to your vision, like where I see Sensible in 5 to 10 years. So Sensible is a partner. We want to be a partner to leading pharma companies that would come to us with their mRNA designs, and we would make them optimized, perfect, high-quality mRNA molecules. And essentially, we'd partner with them, take this RNA through our pipeline, test it on the state-of-the-art assays, and come back to them and tell them, hey, look, here is our mRNA made using our proprietary way. We are, let's make drugs out of it, and then we would manufacture that mRNA for them. So we're very excited about building out the manufacturing infrastructure, and I think this is an area where there is a lot of potential to work on democratization of the biotechnology industry. Moving beyond the clusters of Boston, San Francisco, and London, and thinking where else could the biomanufacturing and kind of bioeconomy be based. And I think that this is really, really, really important. And I will kind of add this last bit. I think that question of mRNA is really a question of technological sovereignty. And just yesterday, I had a pleasure of being at a dinner with where Hermann Hauser was present, one of the founders of ARM. And what really resonated with me was how he was speaking about how chips are literally a part of national sovereignty for many countries, or for many organizations. And this is the way I think about mRNA. Essentially, you should have the capability as a nation to, within certain time period, obviously, be able to produce sufficient amount of your vaccines, of the drugs, of the medicines, in the case of the emergency, and provide for your citizens. And this is the vision for Sensible. We like to think of Sensible as TSMC for mRNA.
That's really exciting. So, yeah, I couldn't agree more. It seems like an exciting future to think about really decentralization of mRNA production. So before we come to an end, I would like to jump on one last topic, and it's fundraising. I think this could be interesting for some of our listeners who are raising for their own startups. What was your fundraising strategy, and where do you stand?
Sure. So I think, again, can't talk about just everything, but we've been very lucky at Sensible that after initial support from Y Combinator and a number of fantastic angel investors like Andrej Kiska, Petr Gajdoš, Martin Rajchan, all of them experienced VCs and support of Civilization Ventures, this Silicon Valley-based early-stage biotech fund. We were able to gather the support of a number of other great funds. The Blue Yard Capital was leading our pre-seed round, together with investing with Zaka Ventures, Amino Collective, and a number of other funds, Possible Ventures, Atria Ventures, other great investors. I think at the time, the big catalyst for our work was the strategic partnership with Ginkgo Bioworks, which has really shown, that, we can get quote-unquote external validation of our technology, and we can spark the interest of one of the leading players in that space at the time. And later on, so that was kind of in early 23, and we were essentially fulfilling our milestones faster than I expected, and moving literally faster than I expected. And very quickly, we were moving to the position where we were ready to raise the next round. And I'm pretty pleased to say that we raised another round that year, led by Recode Ventures, where backed VC with participation from OnSite. So I'll just say that, can't speak about too much about the details. So we are well funded. We are excited about what's coming, and these days and months, I'm really heads down, and I'm focused. I'm really focused on building the technology, building the platform, building the company, talking to the customers, and excited to be raising the next round after we fulfill our milestones, which I'm very confident we will do very soon.
Yeah, thank you, Miro. So with this, I would like to conclude our session. I hope we could give you interesting insights into the future of mRNA and the road ahead in TechBio. Thank you, Miro, for being with us today. Thank you to our listeners, and everyone, goodbye. Thank you.
Thank you very much, and thank you. It's an honor to have a chance to talk with you.
Thank you, everyone. Bye.
That was OnSite Insights, a series of podcasts by OnSite Ventures about deep tech and the people behind it. For more information on our portfolio companies and investment strategies, visit our website, onsite.vc, and follow us on LinkedIn at OnSite Ventures.